Lecture 3 will be on the digestive system. If you're watching this video before your first quiz, just a reminder that this will not be part of that first quiz. Quiz 1 is only on chapters 1 and 2. Okay, so for lecture 3, digestive system, organs of the digestive system fall into two groups. There's the organs from the alimentary canal. These organs start at the mouth. So it's a long tube, and the mouth would be the opening of that tube. Then that leads into the pharynx, which is the neck area, which then connects to the esophagus, then the stomach, then the small intestine, large intestine, and then finally it'll end at the anus. Okay, so it's one long tube. The other group fall under the accessory digestive organs. So these include things like teeth, tongue, and gallbladder, and also digestive glands. So um, these are glands that help the process of digestion by the secretions that they make, and that includes things like the salivary glands, the liver, and pancreas. So here you can see the alimentary canal, which starts at the mouth, so that's the opening, and then it turns into the pharynx, which would be around here, the esophagus, the stomach, small intestine, large intestine, and then it finally ends at the anus. But as you can see, it is just one long tube. It does get wider in some parts, like the stomach, but it really is just one long tube. The accessory glands or accessory digestive organs, so those are the organs that secrete specific substances that help break down the food. So that would be things like the liver, the gallbladder, and also the salivary glands. The digestive process involves 6 essential activities. So there's ingestion. That's when you put the food in your mouth, you chew it, and also the food is mixed with saliva. And some initial uh, digestion is occurring also for some of the food substances. So things like carbohydrates. There's enzymes in your saliva that actually begin the digestion of carbohydrates. But for the most part, it's just breaking down the food, mixing it with saliva, and churning it. Then there's propulsion. That's where the food is pushed from your mouth to your esophagus, and then the esophagus pushes it downward further. Okay, and that's actually called the process of peristalsis because it's a wave-like motion that's caused by the alternating contraction of longitudinal and circular muscles. So you can see peristalsis. Those muscles, they alternate. So when one contracts, the other one relaxes. That's what causes a wave-like motion. It's very similar to the way earthworms move. If you've ever seen an earthworm move, they also move uh, by peristalsis. And it's very similar because they also have circular and longitudinal muscles that they use. And so that pushes the food downward. And it's a very effective process because even if you were hanging upside down and you were eating food, which I mean, wouldn't be, that's something that you would recommend. But if you were doing that, the food would still go down the esophagus because of those wave-like motions that's caused by peristalsis. Okay. And then there's also segmentation. This occurs in the small intestine. So peristalsis, that's from the esophagus. Segmentation, that's the mixing of food, also by contraction and relaxation of non-adjacent uh, muscle, but this occurs at the small intestine. Okay, next is there's mechanical breakdown. So as I mentioned, the mechanical breakdown does begin at the mouth. That's where your teeth are breaking the food items down. And then there's a lot of the mechanical breakdown also occurs in the stomach. So in the stomach, there's acids and enzymes that help break down the food, but the stomach is also churning the food. Then there's digestion occurs in the stomach as well. Um, Absorption, that's going to take place in the small intestine. So the majority of the nutrients that your body absorbs from the food that you eat occurs at the small intestine. The stomach is just there. The main function of the stomach is to break it down and to sort of kill like any bacteria or other microorganisms that are in your food or could be in your food. And then the absorption happens in the small intestine, so after it leaves the stomach. And then there's also defecation. So that's when everything that's digested is already all the nutrients are absorbed from the food items. All the water is reabsorbed, and then whatever is not needed or can't be digested, things like fiber from plants, that's that exits through the anus. So, oops. So here you can also see all these processes: ingestion at the mouth, food is mixed with saliva. There's also mechanical breakdown here. It goes through the esophagus and it's pushed down by those muscle contractions. So the peristalsis, which is the wave-like motion that pushes the food down into the stomach. From the stomach, that's where the a lot of the digestion occurs with enzymes and hydrochloric acid. But there's also some mechanical breakdown because the stomach is churning the food. And then absorption occurs in the small intestine or in the first part of the small intestine. I would say the majority of it. And also water is reabsorbed at this point. And then the nutrients find their way into the bloodstream as they go into the capillaries. And also some things like digestive fats, they end up in the lymphatic system. So the lymph vessels they run very close to the blood vessels. In the large intestine, that's where mainly a lot of the water is reabsorbed. Okay, here. At this point, the nutrients have already been absorbed, so there's really no absorption of nutrients. It's just mainly water. So the mouth is where the food is chewed and mixed with enzyme containing saliva. That begins the process of digestion, and then the swallowing process is initiated. Some of the associated organs include the tongue, the salivary glands, and the teeth. So focusing on the salivary glands, so what they produce is saliva. Saliva is mainly made of water, but it also has some enzymes, and this begins the breakdown of starch with the enzyme amylase. Most of the saliva is going to be produced by the major salivary glands, and these are located outside of the oral cavity. So here you can see the major salivary glands. So there's the parotid gland that's located right in front of the ear. It's the biggest salivary gland. There's the submandibular gland, and then there's also the sublingual gland. The salivary glands are composed of two types of secretory cells. There's the serous cells, which produce the watery secretion, enzymes, ions, and a bit of mucin. And then there's the mucus cells, which produce mucus. So this is why these are all salivary glands, but what they secrete does differ. The sublingual gland is the one that mainly secretes mucus. The submandibular and the parotid salivary glands mainly secrete the watery secretion. The major salivary glands are activated by the parasympathetic nervous system. So when food is ingested, it's going to stimulate the chemoreceptors and mechanoreceptors in the mouth. This is going to send signals to the brainstem or the salivatory nuclei in the brainstem. And that's going to stimulate the parasympathetic impulses um, in the cranial nerves. So cranial nerve 7, which is the facial nerve, and cranial nerve number 9, which is the glossopharyngeal nerve. So as if you remember, the parasympathetic nervous system is involved in resting and rest and digest. So these salivary glands uh, can be deactivated, so it could stop salivation can be stopped if the sympathetic nervous system is activated. So usually during fight or flight. Digestion involves a catabolic process that breaks down macromolecules down into monomers so that they're small enough for absorption to occur. And then there's intrinsic and accessory gland enzymes that are involved in digestion. And these enzymes carry out hydrolysis, where you have water that's added to break the chemical bonds. So in absorption, this is the process that involves the moving of substances from the lumen into the body. So the lumen of the intestine, that's the inner portion of the so if the intestine is a tube, it's the inside of the tube. That's the lumen. So going from there, where the food is traveling, and making its way into the body. So it's absorbed across the cells that make up the small intestine. So cells like these enterocytes, that's where the food is absorbed. It makes its way across those cells and then into the blood vessels, like this capillary here. Okay, but it always goes from the apical region of the cell through the cell and then out from the basolateral side of the cell and then into the bloodstream. There's also these tight junctions that ensure that the molecules pass through those epithelial cells rather than between them. So tight junctions are proteins or protein complexes that are found between cells, and they prevent the the passage of water and solutes between those cells. So digestion of carbohydrates, that's a process that breaks down those large polysaccharides and turns them into monosaccharides that can be absorbed. So starch and disaccharides are broken down to oligosaccharides and disaccharides. Oligo, that means few. So remember that there's polysaccharides, which are it's a complex connection of sugar molecules. Disaccharides would be two sugars. Monosaccharides are single sugars. Oligo is just describing few sugars. Okay, and this begins at the mouth with the salivary amylase. So salivary amylase, that's the enzyme that begins to break down um, saccharide. After after that step, the next step would be for further breakdown into lactose, maltose, and sucrose. So these are also examples of disaccharides. So for example, lactose, that that's made of glucose and galactose. Sucrose, for example, that's made of glucose and fructose. So after the disaccharides are broken down, they're going to be turned into monosaccharides. So these are all the steps that involve the digestion and absorption of carbohydrates in the small intestine. So all of this is taking place in the small intestine. So we have the pancreatic amylase that breaks down the starch and glycogen. So these are going to be the polysaccharides. Okay, these complex uh, carbohydrate molecules, those are going to be broken down to disaccharides. Then they're going to cross the brush border enzymes. So these are located on the apical side of the cells, of the enterocytes. And so border, at the brush border where the enzymes are located, those disaccharides are going to be broken down to monosaccharides, as you can see here. And then those monosaccharides are going to move across the cell. And they're going to exit at the basolateral membrane of the cell. And this is driven by the sodium and potassium ATP pump, because the sodium potassium ATP pump creates a sodium concentration gradient because it's taking sodium out. That means that there's more sodium in the lumen of the intestine than there is inside of the cell, because the sodium is being taken out by the ATP pump. If there's less sodium here inside of the cell and more sodium out here, then the sodium in the lumen of the intestine is going to move into the cell, so down the concentration gradient. When it does that, it brings in the, the monosaccharides. Then the monosaccharides will exit the cell via facilitated diffusion, and then they're going to enter the capillaries. The digestion of proteins begins in the stomach when pepsinogen is converted to pepsin. Um, Pepsinogen is the inactive form of pepsin, and it becomes active when cells that are lining the stomach, that are called parietal cells, secrete hydrochloric acid. That creates a more acidic environment. And so in that environment, pepsinogen is converted to pepsin. So pepsin is what helps break down proteins. Protein digestion and absorption in the small intestine is very similar to the digestion of carbohydrates, because you start here in the lumen of the intestine, and that's where these long chains of amino acids, so these long or large polypeptides are going to be, they need to be broken down to smaller polypeptides. And eventually they're going to be broken down into amino acids. Okay. So we're going to start here at the lumen of the intestine, and that's where those pancreatic proteases, so these are enzymes that are produced by the pancreas. So these are breaking down proteins and protein fragments into smaller fragments. Okay. Then those are going to make their way to the brush border, and that's where the brush border enzymes are going to break down those protein fragments into amino acids. Okay. And it's just like what we saw in the previous process, there's going to be these co-transporters that are going to bring in the amino acids along with sodium, and the and sodium is going to be brought into the cell because of the concentration gradient that's maintained by the sodium potassium ATP pump. Okay, because here sodium is also being, you can see that sodium is still being carried out, so there's less sodium in the cell than in the lumen. So if there's less sodium here, more sodium here, that means sodium is going to move down the concentration gradient, and it's going to move into the cell. And that's going to bring in the amino acid with it. Then it's going to travel across the enteral site, and then those amino acids are going to be carried out, or they're going to make their way out through these amino acid carriers and then find their way into the capillaries. Digestion of lipids begins with emulsification, which is a pre-treatment for triglycerides and their breakdown products. So this involves bile salts, which are produced by the liver, and these bile salts surround or pull apart fat droplets. And when they do that, they facilitate the fat digestion, because when they break down the large fat droplets into smaller ones, that increases the surface area and allows for enzymes to do their work and and complete the digestion of those fat droplets. It also involves these micelle formations. So micelle formations are just products from digestion that become coated with uh, bile salts and lecithin. So lecithin is just a fatty substance. The next step is where lipid products are converted back into triglycerides and they're packaged with lecithin and lipoproteins. When that happens, they form chylomicrons. The chylomicrons are going to be exocytosed from the basolateral side of the enterocyte, and then they're going to enter into the lymphatic lacteal. The lymphatic lacteal, these are structures that are part of the lymphatic system, and they absorb fatty droplets. So these are also located in the small intestine. So eventually, these are going to be emptied into the blood at the thoracic duct because the lymphatic system runs very close to the blood vessels of the cardiovascular system. And the main function of the lymphatic system is to drain interstitial fluid. Um, but when it does that, eventually it does put that fluid back into the cardiovascular system at the thoracic duct. So once in the blood, these chylomicrons are going to be broken down into free fatty acids and glycerol by the lipoprotein lipase. So that's going to be the enzyme that that breaks it down. And you're going to get short chain fatty acids that can diffuse directly into the blood. Okay, so here are the steps for the digestion of fat. So we start here with emulsification. That's when these fat um, globules in the small intestine are going to meet these bile salts. These bile salts are going to surround those fat droplets, or they're going to sort of separate them into smaller ones. By doing that, by making this large fat droplet into smaller ones, it increases the surface area, which makes it easier for enzymes to break down these fat droplets. So we have the pancreatic lipases that hydrolyze the triglycerides, and then they produce monoglycerides and free fatty acids. Then you got the micelle formation, which are going to ferry their contents into the epithelial cells. So then the fat droplets go into the epithelial cells via diffusion, okay, and then these chylomicrons are going to be formed. The chylomicrons are going to make their way out of the cells and then into the lacteals, which are part of the lymphatic system. So the main function of the lacteal is to absorb those fatty droplets. So just to be clear, so here and what's different about this diagram than the other ones is that here we're really zooming out and we were able to see the whole enterocyte. In the previous diagrams, you were pretty zoomed into just the cell, and we saw part of the outside of the cell, which leads into the capillaries. But here we're really zooming in. We're zooming in a bit further. For the digestion of nucleic acids, so nuclei of ingested foods contain DNA and RNA, and there's pancreatic nucleases that hydrolyze nucleic acid to nucleotide monomers. Then there's brush border enzymes that include nucleosidases and phosphatases that break down the nucleotides into the nitrogenous bases, pentose sugars, and phosphate ions. So nitrogenous bases were things like the uracil, cysteine, guanine, and etc. And then these products are going to be broken down and they're going to be actively transported by these carriers, by special carriers in the epithelium of the villi. Vitamin absorption, which also occurs in the small intestine. So there's fat-soluble vitamins like vitamin A, D, E, and K that are absorbed with fats. That's why if you're taking vitamins like this uh, in tablet form, it always says that you have to take it with food or some kind of food that has uh, some kind of fat with it. Otherwise, these vitamins would not be absorbed. Then there's water-soluble vitamins like vitamin C and B. These are absorbed by diffusion, passive, or active transporters. There's also several types of minerals that are absorbed by active transport. One mineral, for example, calcium, that absorption is regulated by vitamin D and the parathyroid hormone. Most water is absorbed in the small intestine by osmosis. The rest of it is going to be absorbed in the large intestine. And water uptake is coupled with solute uptake because it always moves from an area of low solute concentration to an area of high solute concentration. Okay, so we're going to stop here. So for chapter 23, the sections that we went over are sections 1, 4, 10, and 11.